practical or in today theory, we will be focusing mostly on primers. So how we can design a good primer, what should be the specificity and what is the efficiency. But before to go into the practical, I would like to bring two things into the point. First is that what do you mean when you say the efficiency? So when you say the efficiency, it means that it ensures twofold increase. In the base or in the principle of the PCR, we have learned that after every cycle, the PCR should be double. It means the copy number should be double. So in each PCR, if the primer efficiency is good, one copy number will get into 2, 2 into 4, 4 into 8, 8 into 16 and so on, right? So it goes in that way. So that ensures the fold increase. Now, to ensure the efficiency, one must use a target gene with the amplicon, which amplifies around 100 to 200 bases. We will see what it means in the practical session. We also need to ensure that the primer length should be specific, melting temperature, magnesium concentration. The primer should not contain a lot of repeats. It should not contain a lot of run repeats. Run repeats means A or G or C, so something like that. And repeat means like 80 or GC. So that kind of repeats. So this is important to understand. Repeats and runs both should not come into the a good primer. Also, the 3' stability should be ensured because at the 3' end, your amplification starts. And also, it should not form any GC clamps. That we will see in the next slide what it means. So this is about the primer efficiency. Now, what is the primer specificity? When we say, or we have mostly observed, when we do a normal conventional PCR, we get a lot of primer dimer, right? So, along with the gene of interest, you will get a lot of dimers. Or sometimes you will see that there are some non-specific bindings are there. However, if your aim is just to do the sequencing of this particular gene product, you cut the gel and you get your product. In case of real time, when the whole system is dependent on the fluorescence intensity, these primer dimers also contributes, or non-specific binding also contributes to the fluorescence intensity. In this graph, in this picture, you can see that you have your gene of interest in this particular, green arrow. However, at the same time, you also have some of the peaks for dimers or the non-specific bindings. Since cybele green is not specifically going and binding to your gene of interest, it contributes to it contributes to the total fluorescence. So that total fluorescence can be coming from your dimer or the non-specific binding, or it can be coming from your gene of interest. If it is coming from your gene of interest, it is well and good. That is your target. But if it is coming from non-specific binding or the primer dimer, that's a cause of concern. And that is why what we have to first and the foremost look into what kind of primer we have been using for the gene expression studies. I have already said that the amplicon length should be close to 120, but it can range from 80 to 200. Primer length should be around 18 to 20. Melting temperature should be almost 60 degrees Celsius in most of the real-time conditions. However, these values are not hard and fast fixed, but they are very much optimal. And most of the time, if you follow this table, most of the time you will be able to design a good primer. The GC content should be almost 50. If you keep a very low GC content, then it can go and bind at several places. It loses the specificity. At the same time, you have to look, as I said, that the 3' base mostly contains the GC bases, because higher the value, better is the efficiency. So these are some of the parameter which we need to consider. The primer should not cause the self dimer. It means that two forward or two reverse dimers primers should not form a dimer. And also we have to look into the delta G value, that is the free energy, that we will see when we will be doing now in the practical. Also, it should not form a loop. It means that that forward or reverse primer should not self-anneal or form a loop. That will avoid the chance for it to go and bind at the at the place where you would want it to go to the gene of interest. Similarly, heterodimer is something where forward or the reverse primer are going and binding together. So it is different from self dimer where the same primer, two copies of the same primer will go and bind, whereas in heterodimer it is forward and reverse which is going and binding together, right? So these are some of the aspects of the primers which we need to take care. Now, how would you understand that what is the primer efficiency? As I said that ideally twofold increase has been observed in the PCR, and every 10 dilution you would observe 3.3 cycle apart, right? So in this particular example, you can see that first, second and third. So if it is following 3.3 cycle apart, it means that the slope value is close to, in this graph you can see the slope value is close to 3.3. It means that it is having close to 100% efficiency. If not, if it is too, if the amplification curve is less than 3.3 cycle, it means that your primer efficiency is showing more than 100% rate. And if the two amplification curve is showing, or the tenfold dilution samples are showing more than 3.3 cycle apart, then it means that it is not 100% efficient. So basically what we need to understand is that 100% efficiency means twofold rise, and this we will be doing in the practical sessions where we will be doing the curve slope. Another thing is that, so this is how you can maintain the efficiency. To check the specificity, what you need to do is that you have to do the melt curve. Now we know that melting temperature, or when we calculate PM50, it means the temperature at which the 50% of the amplicon is melted, or it's separated, and that gives a peak, and it is a characteristic for each amplicon. So what happens is that if you are getting a single peak like this, it means that it is going and hitting or amplifying only one amplicon, your target gene. In case if it is showing something like this, it means that you have the dimer. So the dimer is also contributing to your fluorescence. In this picture you can see, right? So this needs to be avoided. Another thing you can see is that in this green graph, you will see the fluorescence value is too less. It's almost zero point 0.1 in the y-axis, whereas others are 1.4, 1.8, 1.4. So this means that, and then another thing is that the melting temperature is close to 61, which is too less for an amplicon of close to 100 bases. So this could be only dimer and no amplification. So these kind of things we will be seeing in the file. In this image, you can see that in the higher concentration, if you follow this graph, right, this arrow, you will see that there is a one peak. Whereas as soon as you are decreasing the concentration of the template, so what is happening? You have the more primers or the constant primer, but your target gene is less. So in that case, it is forming a dimer. So what it reflects is that the primer do have the specificity to the gene, but it is either forming a self dimer or the heterodimer. And in case if the template is less, it is forming a loop or forming a self or heterodimer and contributing to the fluorescence. So these all aspects we need to check when we design a primer, right?